Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I have with me Dr. Marc Hedrick, President and CEO of Plus Therapeutics, a clinical stage pharmaceutical company developing targeted radiotherapeutics with advanced platform technologies for central nervous system cancers. Welcome, Marc.
Hi, Nicole. Nice to meet you.
So today, as you probably guessed, Marc and I will be discussing radiotherapeutics and the central nervous system cancer space. However, before we sink our teeth into today's topic, Marc, perhaps you could share a little bit more about yourself with listeners, the journey, if you will, that led you to doing what you do today at Plus Therapeutics.
Sure, Nicole, I'd be happy to. So I'm a physician, been a physician since the late 1980s. So I've been around a while. I was a general and vascular surgeon, then a pediatric plastic and craniofacial surgeon. I practiced at UCLA prior to leaving my academic appointment and going into business. Besides the clinical part of my career, been working in technology development for adults, pediatric patients, and actually the human fetus in development, and both in device and med tech and pharmaceutical and surgical procedure development. So through my career, it's been trying to understand problems that are poorly addressed and trying to come up with solutions for those. So that led me in a team I worked with to discovering novel stem cells in the human adipose compartment back around 2000. And we had the opportunity to develop that commercially, which we did, which led to an academic sabbatical that led to kind of leaving academia completely, getting an MBA along the way, and then ultimately with our board chair building Plus Therapeutics in the 2019 timeframe, and since then we've done 5 acquisitions with the idea that while there've been great developments in cancer therapy over the last decade or more, the improvements in overall survival have been outside of the central nervous system. In other words, the central nervous system cancers those patients are doing about the same as they did 10 years ago. Not much movement there. However, we know that the best treatment in terms of the improvement in outcome, it's not great, but to the degree they're effective therapies, they're radiation related. So the idea behind Plus Therapeutics was to develop novel drug-based, pharmaceutical-based radiotherapeutic strategies to address the central nervous system cancers that have really heretofore not had any meaningful improvements in survival. And that's how we got here. And we've developed a portfolio of drugs, of indications, both therapeutic and now diagnostic, to address those.
Thank you, Marc. That's quite a journey, and a sort of, I want to say a successful one at that, but I understand, of course, these as yet unmet needs within the CNS cancer space. So let's turn now to our central topic and explore a bit more what can be done and what is being done or striven to be done. So radiation has for over a century been considered one of the most powerful methods of destroying cancer. But even now, ways to deliver significant doses to a tumour without damaging normal surrounding tissue are few. And this is where targeted radiotherapeutics come in. Combining innovative formulations with novel administration techniques, a single radiation dose can potentially be inserted into just the tumour or affected tissue until the radiation dissipates and is safely cleared by the body. So, Marc, I was wondering if you could tell listeners a little bit more about this, from the sort of beginnings of this therapeutic space to where we are today.
Sure. Well, let me break that question down in a couple of ways, because the focus here of the company is targeted radiotherapeutics. So let me deal with the radiation issue and then deal with the targeting issue, because both are critical, but both are sort of independent concepts in a way. So big picture, radiation is been commonly used for decades. And I think we estimate maybe 70% of patients that have cancer will get some kind of radiation throughout their treatment course. The problem with radiation as it's been delivered heretofore, and I mean in the last, it's starting to change in the last 5 or so years, but heretofore it's been through external beam radiation. And if you think about an array of radiation going from outside the body into the body through a tumor and out the other side, because you have to go through normal tissues to get to the tumor, but then there's effects after it crosses through the tumor because the radiation doesn't dissipate completely at the tumor level. It really limits how much radiation one can give. And the approach that doctors have used over the years to maximize the amount of radiation they can deliver and minimize the toxicity is to fractionate it. So, for example, I'll use glioblastoma as sort of a little bit of an example of how that works. And those patients, once they're diagnosed, typically get around 60, 70, or 80 gray radiation in terms of the absorbed dose is measured in gray. So they have a maximum amount of radiation that they can give, but it tends to get broken down or fractionated to give a little bit every day for about 6 weeks. So 30 treatments, 5 days a week. So patients coming back to radiation therapy and getting a fraction of the total dose over time to try to build up to a meaningful dose and minimize systemic toxicity. That's a very, it's bad for patients with cancer to come back every day, get radiation. Patients get tired. And while if you look at the survival benefits of various treatments, for example, for glioblastoma, the most improvement of survival is actually from the radiation. But typically these patients will get surgery. They'll also get chemotherapy as well as the radiation, but radiation is the biggest impactor, but the dose is limited in terms of how it's fractionated and the total dose. So that's been the history of radiation therapy for CNS neoplasms and for glioblastoma specifically. So there's a real limitation there, but it works to the degree that, we're doing the best we can with external beam. We're getting the most bang for the buck or the most impact versus other treatments. So I'll cut to the, going to be prophetic here and cut to the chase. So we have a drug that will deliver 10 to 20 times the amount of radiation just to the tumor, not in a beam form, but actually in a pharmaceutical or drug approach. And I'll come back to that in a minute. So the second kind of core element of targeted radiotherapeutics is the targeting. And so a problem with central nervous system cancers generally is that there's something called the blood-brain barrier, which literally exists to be a barrier to toxic substances, to keep them out of the brain and the spinal fluid and so forth. To protect those critical structures. But that also provides a barrier for drugs to get into the brain or the spinal fluid and so forth. It's only about 2% of all drugs get a meaningful concentration of the compound, the active pharmaceutical agent, into the central nervous system. So from a targeting perspective, if you have a targeting strategy that's based on a molecular approach or pharmaceutical approach, you still have to deal with this issue of the blood-brain barrier, which is very thorny and very difficult to address. So there are other ways to target besides molecular targeting or an intravenous drug that has a molecular key and lock mechanism to go right to the tumor in the brain. And that's a direct targeting. And we took the approach early on that we think that we can get the most amount of radiation through direct targeting. And what do I mean by that? So there's a technology called convection enhanced delivery that was developed, I believe, at the NIH maybe 30 years ago, that uses a pressure-based infusion technique. I think about it like biological fracking, if you're aware of the oil and gas industry now. Oil and gas is produced in shale deposits. You subject the fluid to pressure, it moves through the ground, breaks up the shale deposits and yields the gas and oil. we're doing the same thing in the brain, although at a much kind of different level and different pressure level. So we actually frack the drug into the tumor in a very precise way to cover the tumor in the other region of interest, but spare the normal tissues. That's how we get such high doses of radiation to the tumor region of interest, but spare the normal tissues. And that's how we can turn the dose up by 10 to 20 times. So that's one way of addressing a direct targeting of the radiotherapeutic without and essentially parachute beyond the blood-brain barrier and get to the tumor. A number of other tumors affect not so much the brain substance, but they affect the fluid around the brain. And in fact, there's an epidemic right now in the US of a cancer called leptomeningeal metastases. And that's a metastatic complication of very common cancers where the tumor ends up in the fluid around the brain. Well, there's actually a chemotherapy port called an Ommaya reservoir that can be placed under the skin into the ventricle, which is part of the fluid-based system, and allow literally two-way access to the brain fluid, both for removing fluid for diagnosis, but also delivering drugs. It's called intrathecal delivery or delivery into the thecal space or the CSF space. So we can sort of take a step back and say, okay, is the tumor in the brain, spinal cord, and we have this technology called convection enhanced delivery. There are now commercial systems made by big companies to address that. And then there's a common procedure called placement of Ommaya reservoir, which allows access to the fluid. So kind of to tie it all together, the radiotherapeutics are drugs. They give super high doses of radiation just to the tumor. The kind of radiation is important, and we could talk about that, but it gets pretty technical into the physics. But the delivery, I think that a direct targeting approach is better for the CNS versus a molecular targeting, which is typically used for things outside the brain and spinal cord.
Thank you for that overview, Mark. And please do feel free to go deeper into the physics if you wish. A majority of our listeners perhaps could very much keep up with you if you wish to share more in that regard.
Sure. Well, let me. So part of the development of radiopharmaceutical drugs for the central nervous system, there are a couple of issues that are really important just from the drug development perspective. The first, and it's more physics heavy, which is the selection of the radioisotope. So the first radioisotope that was really used en masse for patients was I-131. And that's preferentially taken up by the thyroid and is still used today commonly. And that has its own sort of physics. And when I say physics, I mean things like the amount of energy delivered, the path length of the radiation, the decay cycle. Does it produce sort of downstream radionuclides that might have different effects as the one that you're delivering initially? Is it gamma particle? Is it alpha particle? Is it beta particles? The different types of radiations that are delivered. The radiation density can change. The chemistry of the radioisotope can be relevant. So we've elected to use a radioisotope of rhenium called rhenium 186, the 186 isotope of rhenium, because the combination of those factors, path length, energy, it has both a beta and a gamma particle, which allows real-time imaging. We can measure the beta particle, which is a high energy electron, causes the cancer to die. And the mechanism of action is well known. It's double-stranded DNA breaks. The body's normal repair mechanisms can't overcome those. And it preferentially treats fast-growing cells like tumor cells, but also has a gamma particle. So 1 out of every 10 particles that are emitted from rhenium is a particle that we can image. So you think about it, you take an aspirin or any other drug, you take it, goes in the circulation, and one can't tell on a per patient basis, hey, where is that drug and how much of that aspirin is getting to my headache? You don't really know. You just know that 80% of the time you take an aspirin, it's going to make your headache better. But 20% of the time it doesn't, you don't know why necessarily. We can actually take our patients after they've been treated, put them in a scanner called a SPECT CT scanner that measures the gamma emission and can show you where it is in the body, from, the central nervous system to, other organs and tissues like the bone marrow and other things where there might develop toxicities from the radiation potentially. And it can also tell you how much radiation is being actively delivered to the tumor. And so from a drug development perspective, it's a really powerful system because we can know on each individual patient how much radiation we delivered, how we delivered it, where was it delivered, and how much radiation was delivered to the tumor, and correlate that with outcome. So it's in a way, it's our way to get pharmacokinetic data. So the physics of the radioisotope become very critical in terms of the outcome and the effect on particular tissues. So that's kind of one part of this. The other part is the drug formulation. So radioisotope does the cancer killing and the imaging and so forth. But then how does one formulate it? And that really gets more to the targeting strategy or what you're trying to do pharmacokinetically. So there was one drug approved back in the late 1990s for lymphomatous meningitis, which is lymphoma of the central nervous system fluid. And that drug was a encapsulated, liposomal encapsulated chemotherapy called cytarabine. And it was the purpose of encapsulating it is it was a depot version of the drug, and you could put it into this central nervous system fluid and with the goal of improving survival, but eradicating the tumor in the fluid and improving the outcomes. That was pulled off the market a number of years ago for business reasons, not for safety reasons or the fact that it didn't work, but it approved by the FDA. That's now off the market. So we've taken that strategy and taking it one step further. So we encapsulate rhenium, which is no small chore to take a very powerful radioisotope, put it in a lipid envelope, and then deliver millions of those into the thecal space. And what it does is it protects the drug from getting consumed by the body's macrophages or go into the circulation. So it's a depot version. So we can treat a patient with a single injection of the drug, whether it's for leptomeningeal cancer or for pediatric brain cancer, adult brain cancer, and so forth. And then have the drug stay where you want to put it for its entire decay cycle. So for example, rhenium-186, the half-life is about 90 hours. So it's around after injection for several weeks, but it stays where you put it by virtue of how we deliver it to the region of interest, elaborates all of its therapeutic radiation to the tumor, the region of interest. And then by the time it's absorbed by the body, it's substantially decayed so that there are no untoward effects or effects that cause complications or adverse events that go to the rest of the body. So the drug formulation becomes very important. And then marrying it to the proper radioisotope are kind of critical elements of the drug development process to get to an agent that can potentially be successful clinically and commercially.
Absolutely. I mean, it's fascinating science, and thank you for sharing all that with us today on this episode. So there we have it, not quite in a nutshell, but in an explorative fashion, how leveraging novel drug formulations and technologies, this method can deliver a single, safe, and effective dose of radiation directly to the tumor. And this works in both adults and children, I believe, Mark, those who are battling rare and difficult to treat cancers. And we've had, as you've been giving us examples along the way, such as glioblastoma and also LM or leptomeningeal metastases. And indeed, I believe also Plus Therapeutics recently received FDA orphan drug designation for Rayobic, I'm sorry if I pronounced that wrongly, for the treatment of LM in patients specifically with lung cancer. So I was wondering if you could share more details about that, Mark, and also if you wanted to provide any other examples of the use of radiotherapeutics in the rarer and, as you have been doing, the specifically CNS space.
Sure. So almost by definition, any cancer of the central nervous system is a rare disease and qualifies for orphan drug designation. Oftentimes, these are unmet medical needs. For example, for leptomeningeal cancer, there's no drug approved currently for it, and doctors take a kitchen sink approach. But half the patients, once they're diagnosed, go to hospice. because the treatment options are so terrible. There aren't good solutions out there. So orphan designation in and of itself is helpful in the drug development process. But our goal from a just prior to approval is breakthrough designation and other FDA regulatory opportunities to speed development. Let me talk a little bit about the epidemiology of these different cancers and what we're going after and why. So for adult types of cancer, both in metastatic and primary cancers of the central nervous system, the way to think about it is there are primary tumors and there are metastatic tumors. Primary tumors are actually extremely rare. So I mentioned glioblastoma in the US, and just use that as to kind of anchor the numbers here. There are about 15,000 patients a year that get glioblastoma. Very small number of patients. It's such a devastating disease, and essentially all patients die from that disease. Some sooner, some later. That you talk to anyone, they know somebody that's had it, or they have a friend of a friend that's had it, and there's no hiding it. People aren't walking around necessarily with glioblastoma without knowing that they have cancer, and it's fairly obvious. And it's no two ways about it. It's a horrible, devastating disease with a horrible prognosis, but it's a very small number of patients. And it has elements, it's tumor biology. There are parts and things about it that make it very difficult to treat. It doesn't metastasize outside the brain. It basically, patients succumb because it directly invades into the brain structures adjacent to the primary tumor and can do so very quickly. And so eradicating not only the tumor, but the area around the tumor is critical. And that's sort of a predicate for our approach. You think about glioblastoma, it's like an octopus with tentacles. And to get long-term survivors, you have to treat the tumor, which is the body of the octopus, but you have to identify the tentacles and treat those with radiation doses. And we know empirically that's over 100 gray of absorbed dose. But glioblastoma, primary tumor, very rare. The tumors in the brain tissue, the most common, are actually metastatic cancers, and those come from much more common cancers, things like breast cancer, lung cancer, melanoma, gastrointestinal cancers will metastasize to the brain substance, so the brain tissue itself, or they'll metastasize to the fluid around the brain. And there's separate pathologies that develop depending on how the metastases go to either the brain or to the fluid. But to put it in perspective, there are about 400,000 patients in the US that have metastatic brain cancer, and then about 400 to 500,000 patients a year, it's substantially underdiagnosed, have metastatic cancer to the fluid space around the brain. So the numbers really dwarf what you see in primary brain cancer. So you kind of look at the pediatric population, those are about 1/10 as common as adult primary brain cancers like glioblastoma. So that all sort of puts it in perspective. Now, leptomeningeal cancer is a very interesting one because there's an epidemic of this in the US, and there's an epidemic because there are increasing numbers of drugs that do a really good job of local control of the tumor, whether it's in the breast or the lung. They maybe don't eradicate it. There's still tumor cells, but they activate the body's immune system, for example, to keep the tumor in check. And patients live for many years, but there's still tumor cells there. And those cells can get into the bloodstream. And then one cell theoretically could get into the fluid space around the brain. And then once it's there, it's protected. It takes advantage of the blood-brain barrier, just like the brain and the spinal cord, normal tissue, take advantage of it to keep poisons out. Well, it keeps drugs out too. And there may be 500,000 patients, based on autopsy studies, have this. And as I mentioned, there are no approved drugs. And that once there, about half of these patients go to hospice because there's nothing approved. And there's sort of a therapeutic nihilism that is in the oncology community right now with respect to this disease because there aren't good treatment options. And the ones that are treated, they only live about 4 to 6 months, and doctors sort of, throw everything at them. So there's a real sizable number of patients that have no treatment options that we're in phase 2 dose optimization. And I think we'll be able to move that very quickly through FDA if the data continues to be, very encouraging. It is right as it is right now, getting very high doses of radiation to the region of interest, to the tumors. Single dose, it gets 20x what you can give with external beam radiation. And even external beam is not great for this because it tends to be more disseminated throughout the fluid. So there's tremendous commercial opportunity here, as well as of course, the key opportunity of extending life in these patients. We've had a number of patients that live several years with the tumor, if we can catch them early and treat them with multiple doses. So that's kind of that landscape epidemiologically and kind of where we're focused. And then it's hard to fully talk about the opportunity therapeutically and commercially without talking about diagnostics. And there's an equal problem to the therapeutics of these kinds of cancers that lie in the diagnostic problems with these patients. So while one can clearly image a brain cancer with MRI or CAT scan, it requires a brain biopsy to tell you what it is. And you can go in with a needle in a highly precise way and confirm the diagnosis. And that's in fact what happens with our treatment for glioblastoma. A patient goes in, there's a lesion there, they get a biopsy, and then at the same time they get up to 5 catheters placed in and around the tumor. It's all planned out in silico beforehand. The neurosurgeon follows the planning template, leaves the catheters in, the drug's infused the next day, and then the patient has the catheters removed and in their room, and then they go home. That's the treatment for glioblastoma. For LM, we use the chemotherapy port called a Maya reservoir, as I mentioned a minute ago. But the problem with cancers of the fluid is they're diagnosed very late. MRI is very poor and nonspecific. These tumors aren't like a golf ball sized tumor, for example. They're like BBs of tumor or a layer of tumor that can be very difficult to see by imaging. So imaging is not a great way to make and confirm the diagnosis. And there's no biopsy that's possible. You can't, it's very difficult to even identify the lesions, much less get some type of needle device that just to get tumors doesn't exist. Now, you can sample the fluid from around the brain spinal cord with a lumbar puncture. The problem is that the diagnostics for that are the same, kind of circle back to where we started when I was in medical school in the 80s. And it goes even back farther than that. It's things like cytology, which the answer you get is there's tumor there, there's not tumor there, or we don't know. We can't tell. It's only about 50% sensitive. Nowadays, that's a very low sensitivity. And you look at protein and glucose, and there's sort of indirect clues to whether there might be cancer there, but that's far from being a definitive diagnostic. As a result, patients that come in with maybe a history of breast cancer and maybe some neurologic changes, it can take a while to get to the diagnosis and multiple lumbar punctures. And then those analyses don't tell you what kind of cancer it is and don't tell you how many cells are there. You can't quantitate the tumor like you can with an MRI. So we actually acquired a technology from a US public company that has a highly sensitive, highly specific diagnostic platform. And we think that will double or perhaps quadruple the efficiency, and I mean sensitivity and specificity for diagnosing LM. And what that will do is, with the few drops of the fluid from around the brain, we can tell how many tumor cells down to maybe 1 cell per 8 mLs of fluid. We can tell what kind of tumor it's from. We can interrogate those cells for expression patterns, both intracellularly and extracellularly. For example, for breast cancer, is it HER2 positive or HER2 negative? For lung cancer, what's its KRAS status, or what's its PD status on the extracellular membranes of those tumors? And then we can assess the cell-free DNA. So we can provide enormous amounts of data from very small amount of fluid that help you make the diagnosis, or more importantly, rule out the diagnosis. And that's a relief to patients to know that they don't have the disease. But then say, okay, what are the best therapeutic approaches? What kinds of drugs might work or not work? And is the tumor getting better with treatment? Are we decreasing the number of tumor cells? Or is it actually increasing and maybe the therapy is not working and we're not seeing a response? So we're a unique company in that we have leading technology as it relates to treatment of CNS, primary and malignant neoplasms, but also best-in-class diagnostic for this. So we're actually relaunching that diagnostic in the next month or so. That was actually commercial. We acquired it, tech transferred it, and now that's up and running in our centralized laboratory in Houston, Texas. And from there, we can essentially test, all patients in the U.S. that may have suspicion for the diagnosis of LM, and then provide ongoing data to physicians to make treatment decisions for those patients.
It's just amazing, Mark, and thank you for sharing that with us. So all things discussed today with this sort of combined technology for treatment with drug formulation, with diagnostics, the whole 360, if you will. My last question, as listeners know with these podcasts, looks to the future. So if we consider the future of the radiotherapeutic space and the future of this rare disease CNS cancer space, what's your, and it seems a very flippant statement right now, what's your crystal ball prediction, given the gravity of what we've been discussing, for the future use of this to address these unmet needs?
No, thank you for the question, Nicole. So the way I'd look at it a couple of ways. The first thing is, I think radiotherapeutics, so using a pharmaceutical approach to deliver radiation, will supplant to a large degree external beam radiation. There may be some opportunities where they could be combined, use of external beam and radiopharmaceuticals, but I think it will supplant them because the value proposition is so compelling. And I won't go back over that, but I mentioned kind of why I think the value prop is there. So I think that's prediction number one. Prediction number two is that, and this data was presented in Vienna, Austria, I think in the last month, by Dr. Andrew Brenner, who is a principal investigator on our LM trial. He's showing that the use of a radiotherapeutic like Rheobic, our drug, in the cerebrospinal fluid actually activates the innate immune system and may make drugs that are currently available, take an immuno-oncologic approach to those tumors, might be synergistic. So I think there's a strong suggestion in that data that combinatorial approaches will give the best outcomes. I think that's sort of almost predictable in a way, but now we have data that shows that, that's actually real. And then finally, I think there's some room for combining direct approaches and molecular targeting, as well as tweaking the formulations and also the delivery technology.
Thank you, Mark. Well, it certainly sounds positive to me. Thank you for your insights today. It's been a pleasure.
Thank you, Nicole.
And that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on Apple Podcasts, Spotify, Overcast, Pocket Casts, Podbean, and pretty much wherever else you download your other podcasts from. Just search for pharmaphorum and follow and hit subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us on Blue Sky and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.